Okay, I think we will start right now. Hello, everyone, and welcome to our LIF webinar. I am Anastasia from Tissue Diagnostics, and I will moderate today's session. We are glad that you are here and that you're interested to learn more about our newest version of the image analysis software, StrataQuest. And today, Sanchit Seth, our application specialist from Tissue Diagnostics Australia division, will showcase the software capabilities on a colon sample stained for several immune cell markers. You will hear from him very soon, but first, few organizational moments. So for question, please use the Q&A box. You can ask your questions anytime. They will be answered in the end of the session. If we won't be able to answer them straight away, we will contact you via email and follow up. For moderating purposes, chat is disabled, so please use Q&A function throughout the meeting. Also, the meeting will be recorded and the recording will be sent to all participants after the meeting. So that said, Sanchit, please, the stage is yours.
Thank you so much, Anastasia. Hello, everyone. And you can see my screen and I'm audible to everyone, right? Okay. So hello, everyone. I'm Sanchit, application specialist from Tissue Diagnostics Australia division. In today's session, we will be exploring the StrataQuest 8 through the spatial immune landscape on a colon sample. Basically, this session will be a mixture of a couple of slides of a presentation to give you an overview on what different features are there for StrataQuest, and then we will take a deeper dive into the applications using one of the bright field sample and the fluorescent sample. Without any delay, let's start with what to expect from this session. So we will be participating in a live demo. As I mentioned, that we will be going through a couple of the samples. And obviously, we can have a Q&A sessions on a later stage, depending on if you have any very specific analysis requirements. And if you have a question that if it is possible to do such analysis with StrataQuest, we can have those kind of questions and answers. And later on, learn the basics of the image analysis and navigating through StrataQuest 8 interface. So we will be seeing how the StrataQuest 8 interface actually looks like and experience practical samples of spatial immune cell analysis in colon tissue. Explore among other exciting tools like tissue classification and phenotypic interaction. So we will be going through, some of the most used tools in StrataQuest. Learn how to use the tools for data mining. So we will be seeing how you can create the Disney plots, UMAP, song, and other diagrams. So how you can actually see your results in a more concise manner and how you can export the images and how you can create different image overlays and how you can get the data ready for the publication. So the first question is, why StrataQuest? How we are different from any other image analysis platform that is present in the market? The very first thing is it is built by the scientists for the scientists. So we do not outsource our software development to any other third-party companies. So we develop our own image analysis software. So we have a team of researchers as well as scientists who do the development of the software. So it is developed in-house by the team of researchers. And the next point is one platform, any image type. So StrataQuest is a platform. It's not just one tool. It is a platform which can be used for the analysis of bright field images, fluorescence images, multiplex images, multispectral images. So all of those images can be imported and analyzed for different kind of requirements of analysis under the same umbrella of StrataQuest platform. So flexible, the next point is flexible for every user. So StrataQuest is equally powerful for the beginners and experienced core facility managers. We will be seeing this today in the live samples as well, that in StrataQuest, you can create your own workflows depending on the requirement of the researchers. At the same time, we also assist the researchers to develop their workflows, and we provide it in an easy mode. We call it as StrataQuest apps. So we will be seeing that how you can use those apps as well. And the next point is versatile installation options. So StrataQuest is, as you might be aware, that Tissue Gnostics has a Tissue Facts platform and a StrataQuest platform. So we have this as a whole setup which can run, which can be integrated between the scanner and the analysis platform. But the StrataQuest platform can be run standalone. So you can not only use the images that are scanned using the TissueFacts platform, but we support other file formats like Zeiss file format, Olympus, Akoya, Hamamatsu, NanoZoomer, and multiple other file formats that can be imported directly in StrataQuest. You can also import the generic file formats like TIFF files, TIFF multi-page, or very basic file formats like JPEG or PNG files as well to do the image analysis. And last but not the least, export support and continuous improvement. It links to my first point that it is built by scientists for the scientists. So tissue gnostics always, keep the support team as a first priority. And then we are always available for the researchers for their innovative and new research questions. And we develop custom solutions upon request as well. With that, let's get started into the StrataQuest platform and have a look at a couple of the samples that I have prepared for today. Just give me a second. So once you open StrataQuest platform, how it looks like is this. Once you import the image in StrataQuest, this is how it looks like. We can divide StrataQuest into basically three different compartments. The left-hand side, in which this is the place where you can have the information about your samples. In this case, it was just one sample loaded, so you can see just one sample here. And on the top, you can see the output images of every engine that you will be running or every image processing you will be doing. On the right-hand side, you will see four different tabs. So images, it's where you can create your own image overlays. Like, if you want a background image as an original image, or if you want a segmented image as a background image, and you can create your overlays in the images tab. We will see how we will be using it. Diagrams is where your data will be plotted in the flow cytometry manner. We will be going through it, how you can create the diagrams and how it is useful to actually summarize your image analysis. And you can actually look at the analysis and visually evaluate what the analysis is telling you. Object inspector is you can, after the segmentation is done, each object gets a unique ID and you can track backward and forward using that unique ID, the measurements that you have done for that particular object. Raw data is basically you will be seeing all the data, whatever you have analyzed within the image analysis workflow under the raw data section. In the center, what you see is the image. So when you load the image, your image is presented in here. You can zoom in, zoom out like in Google Earth. Zoom in into a single cell level. So you can navigate like in Google Earth in here. There are some really cool tabs or features in underneath where you can create your own region of interest. So if you want to create an ROI, you can create an ROI like this, and then you can do the analysis within this ROI, and then this ROI analysis can be linked with the actual sample as well. So there is other features like you can view your map of the image. So if you zoom in, zoom out, you can see the map here. You can navigate through the image from the map. You can have annotations taskbar here. Annotations can be done in your sample. And one of the coolest feature is this exclusion area. So sometimes when you do the image analysis, what you see is your certain parts of your sample is either oversaturated or the staining has not worked in those areas, or maybe just the tissue has folded during the processing. So using this exclusion area, if we say pre-drawn, what we can do is you can go to that particular area and draw this image. On the image, say exclude. So you can do this exclusion for that, just give me a second. So you can have you can have this exclusion area created. So what will happen is all the analysis that are done, it will be done within this image, excluding the any of the data that is available in here. Like in this case, this analysis was already done, but as I created this exclusion area, we saw that all the analysis has to be done again because it has removed this particular section, the analysis what are in this particular section. So we can delete this exclusion area and then let's rerun the sample. And you can see when the sample is run, you can have on the taskbar where all the summarized data will be there that your sample is running and all the samples, if there are multiple samples in a project, they can be aligned in the list and they can be seen here. On the left-hand side, coming back here, this is where you can get the information about your sample. And this is where you actually create your analysis. So in StrataQuest, we perform the analysis in layers. So what that means is basically you can create, if you have a research question, like in this case, it is a colon sample with Ki67 marker for proliferation. And the obvious question here was how many cells are proliferating or how many cells are positive for Ki67 and where those cells are present. So if you have questions like these, you can break it down into different smaller, bite-sized questions. Like first, if you want to know where Ki67 cells are present, you can actually see you have to identify all the cells first, and then after identifying all the cells, you have to see where those Ki67 cells are present. So if these are these two questions, so in layers editor, we said, okay, we have a layer called nuclear segmentation and we have a layer called classifier. You can have as many layers as you want, depending on how complicated your research question is. And what you will see is every layer has a same sublayer structure. So so every layer has a same sublayer structure, which includes of 6 steps: pre-processing, detection, post-processing, manual correction, measurement masks, and measurements. So these are the 6 steps that are required to answer your research question. So, but all these 6 steps are not compulsory to use. They can be used as and when required. Like if your image has a lot of background, you can do some pre-processing, say background removal under the pre-processing step. And following that, you want to identify all the nuclei. You can go into nuclei, you can go into detection, and you can see there are different detections, like for RNA-scope or fish analysis, we have dot counter, we have nuclear segmentation, classical nuclear segmentation, and then we have nuclear segmentation, DNN-based nuclear segmentation, and then you can do area measurements as well here. You can do these kind of detections under the detection tab. And the post-processing is where you can actually do some of the post-processings, like remove objects that are less intense or way more intense, or you can remove objects that are smaller than certain area or larger than certain area. Again, it is optional to use, but it is available if you require it to be used. The next step is if the algorithm identifies something that you think it's not identifying it correctly, we also give this flexibility and the power to the user that you can manually look at the results and see, okay, this is not my actual nuclei, this is over segmented, this is under segmented. So you can do manual corrections from here and you can draw within the manual corrections. After that, the tab measurement masks is here where you can create the secondary measurement structures. In StrataQuest, how we start doing the analysis is we use nuclear channel or nuclear marker as the master channel to identify all the nuclei and to identify the markers that are expressed in the nuclei. So if your marker is expressed outside the nuclei in the cytoplasm or in the membrane, so what we can do is we can create a secondary measurement mask around the nuclei, and you can create those masks from here, like cellular mask. If you wanted to use the grow operation, we will see it in this sample, how this grow operation is used, or if you want to identify the centers of each of the nuclei identified. In the measurement section, if you see, there are multiple measurements that can be that can be done. Like if you are doing the dot analysis, you might use this particular operation where you can count number of dots within your nuclei or within your cytoplasm or within the secondary measurement mask that you have created. And then the most used one is the standard measurements. So if you want to do the area measurements, if you want to do the mean intensity, so by default, we have provided these many parameters. If you can have a look here, if you want to look at the compactness of the cell or perimeter or sum of intensity, minimum intensity, then percentiles. So you can do these measurements uh, if you want. It is available for you, but again, it is not compulsory that you have to do all these measurements. Now, coming back to this particular sample, where it is a colon sample, the question was to identify the Ki67 marker in this. Because this is a bright field image, uh, and most of us know that the image analysis happen on a gray image, we need to identify the gray image for the nuclear master channel. In that case, we will be using pre-processing. And if you go here, there is an option called color separation, which assists you in separating the blue versus brown. So it is not limited to just these two colors, blue or brown, but what it does is the color separation, it extracts the RGB values of hematoxylin and RGB values of the Ki67 marker in this case. So it automatically detects your stains which are present, like in marker one, it's blue and brown. Once you analyze it, what you see as an output is the blue channel for the nuclei and the brown channel for the Ki67. Now we have identified the nuclear channel, which is the blue channel in this case. We can go to the next step and say, identify the nuclei using the blue channel. So in this case, we just selected nuclei. And once you selected that, these are the options here. And once you set the options, these are the default parameters. And what you get as an output for this is you'll see a nuclear segmentation here. So after the nuclear segmentation is done for this one, the next thing was we didn't have to do any post-processing because we were happy with the nuclear segmentation in this case. We did not do any manual correction. We did not create any measurement masks because Ki67 is a nuclear marker, so we do not have to create a secondary measurement mask. We directly went to the measurements part. In measurements, we used nuclei, segmented nuclei, and the blue shades to identify the mean intensity and the area of all the nuclei. Similarly, we used nuclear segmentation because it is a nuclear marker. We have selected a nuclear segmentation here and then brown shades to identify how many of these markers, how many of these cells are positive for Ki67. We selected area and mean intensity here, and then we just plotted these diagrams. So now coming to the diagrams, this is what is, we like to call it as a USP of StrataQuest. So we not only assist you in doing the image analysis, but we also assist and provides tools for the researcher to actually look at the data, what the data is telling you. After the nuclear segmentation is done, each individual nuclei gets a unique ID. Segmented nuclei gets a unique ID, and that's how you can actually see what the data is telling you in here. In this scatter plot on the right hand side, you can see there is nuclear brown shades mean intensity and nuclear brown shades area. That means nuclear area versus nuclear intensity for the brown marker. So what happens is, because we give this unique ID to each of the identified nuclei, these dots represent in the scatter plots, actually, they're not arbitrary dots, but they represent individual cell in the image space. And because of this unique ID, you can basically click on any of the nuclear segmented, and it will start blinking where that particular nuclei is present in your dot plot. That, like in this case, it was a brown positive nuclear. It is blinking above the cutoff. It's a brown negative or a blue positive. It is just in the blue below the cutoff. So the image analysis in this using the StrataQuest actually helps you to evaluate what your data is telling you. It just does not give you the Excel sheets or just the data points, but you can actually see what your data is telling you. So because we click on the cell and it is blinking in the dot plot, we are going from image to data. We call it as forward connection. And the other way around is also possible. So we have set these two cutoffs over here. And if I just say view backward data for upper right quadrant, you will see all the Ki67 positive nuclei. They have been highlighted in a different contour. And this is how you can actually see if your cutoffs are right, what the data is telling you. You can actually, dig deeper into the spatial environment of the data, that where these cells are present, how these cells are behaving with each other. And then another very valid point, or very, what I like to call it as a cool feature, is once you have created this backward gate, you can actually just right click and say save image with displayed, save displayed image with data. And this particular image will be saved for your publication. It is ready to go for your publication or your, weekly lab meetings. So now you don't have to, take the images from or create a screenshots of images and take it to PowerPoint, start drawing arrows that this is my positive area, this is my positive cell, this is my negative cell. All that can be done using StrataQuest just with this backward connection. So you can export your data directly and this image will be saved, can be saved in TIFF, JPEG, PNG, and multiple other file formats. Now, taking this image a step further, we can close this backward connection. The next question was, okay, we have identified all the Ki67 positive nuclei in this case. What if I want to just see the nuclei that are present only in this villi region? So in this case, we used another layer. That is the second research question. So we created another layer called classifier. In this case, we did not do the nuclear segmentation again, but we went one step further. So you can rename the layer. We named it classifier. Under pre-processing, instead of color separation, we chose classifier from the list. So if you see here, we chose classifier. Depending on what your image requires, there are a lot of different features or tools are present, which we call it as engines, which can be used for your image analysis. When the classifier was loaded in the layer, we can just create, train the classifier saying, yeah, this is my lumen region, this is my stroma region, this is my background. And it runs a training starting from in this case, it's pretty much instant because the image size is small, but the training also depends on how much training you have done or how big is your sample. It can vary from somewhere around instantly to few minutes kind of thing. So once the training is done, what you get as an output is, just show you here, close this down. It's like this. So it has identified these lumen structures and then the stroma and the background. And after this, what we see is because we wanted just this layer of cells to be identified, what we did was we took these lumen structures and we identified the area of these structures. And after the area was identified, what we did was we used a measurement mask. As I mentioned, that this is optional. This was optional to use. In the previous layer, we did not use it. But in this case, when we use the grow operation, what we set as a grow, you can set a parameter, say, okay, 8 micrometers. And we use this area for the grow operation. And what we get as an output is the mask itself has grown a little bit. just covering the single layer of these cells. Now, what we can do is, because these layers are, they're related to each other, they can talk to each other. So you can export the data of one layer and import it into the other layer. So we wanted to take this data and we took it into the nucleus segmentation layer. We imported it from the classifier layer, grown mask. And then what we did was we created another scatter plot on the right-hand side saying which divided our cells into two different categories. One which are in the luminaria, which are in the villi region, the cells, and the other ones that are present in the stroma. And then you can create gates like in flow cytometry, and then you can just say view backward gate for brown glands. And now you will see only the brown positive nuclei that are present in the villi region are highlighted in brown, whereas this particular nuclei, which is positive for Ki67, it is not highlighted because we have selected it just to see where my Ki67 cells are present. And then if I click on this one, you'll see this is present in the other category. If I click on this one, this is also in another category. So you can actually evaluate how your data is looking like, and you can export all this data from the, if I click on any of these scatter plots, you will see the data for all the scatter plots in here. And then you can just right click it and export the data, export data for this particular scatter plot into an Excel sheet, or else you can actually export this particular scatter plot as well. And you can export it as, PNG or any of the file formats. So these scatter plot can be used directly for your publications. That is the summarized data for your analysis. But if you are really interested in the raw data that you want to look at and do some downstream analysis of your own, you can double click or just go into raw data. In this case, I have just opened it, and now you will see that the event label, this is what I talked about, that the unique ID we give it to the each and every identified or the segmented nuclei. So you can see there are total 2056 nuclei or the cells identified. And then you can get the area values, intensity values, all the measurements that you have created in this raw data. And this can be exported as well for the further downstream analysis. So with this, I think I could show you some of the basic structures or some of the basic analysis that can be done with StrataQuest. Now, taking it one step further, as I mentioned, that we can use the same platform, but the same platform can be used for bright field analysis as well as fluorescence analysis as well as multiplex analysis. So the next sample that I have here is another colon sample, but in this case, it was a fluorescence sample. It was a 5-color marker. And in this case, they just wanted to see the immune status of different cells. There were CD4, CD8, PD1, pan-CK for epithelium, and DAPI for nuclei. So these were the 5 markers that were present. And again, using the similar kind of workflow, we have created some of the analysis for this. But in this sample, I would like to show you that when I presented in my presentation as well, that we provide solutions for the beginner to the people who or the researchers who really want to do their own analysis or create your own workflows. In this case, we have the workflow was created in a very similar way, like in layer structure, but it has been condensed down into what we call it as an easy mode or an app mode, where it is just reduced to some parameters that needs to be changed for your image analysis to be done. So you can do you apply this app for say hundreds of the samples. That doesn't there is no limit on that, but you don't have to even deal with the layer structure if you find it complicated. So you can have a controlled parameter kind of structure. In this case, we wanted to identify all the nuclei. We wanted to identify the structures like stroma, background, and the epithelium structure. So in parameter section, instead of the layers, you can see classifier. When you click on the classifier, again, background, epithelium, and stroma, you do the training similarly like what we saw in the other sample. Once that is done, what you get as an output is, show you, output like this. So, the green isn't for the stroma, the pinkish color or the reddish pink color is for the epithelium, and the gray color is for the background. After that, if you wanted to identify the whole tissue, you just set here some parameters, you can identify the whole tissue. When you want to do the nuclear segmentation, so you can actually go to the nuclear tab, put the input image, in this case 425490, it was basically the DAPI channel. We input that, we set some of the parameters here, and you can see the nuclear segmentation done for the whole sample. Similarly, when you want to do the cell masks, you don't have to again go to post-processing steps. You can just go into parameters. You want to create a cell mask for CD4, PD1, CK, and CD8 because all four of them are cytoplasmic markers. So what you see is basically epithelial cells identified for the CK positive ones, and then cytotoxic T lymphocytes, helper T cells, and you can create distance zones. So if you go to the proximity areas, you set your distance zone, say 25, 50, 75, 100, 150 to identify which of the phenotype is present, uh, present next to the epithelium region in this case, or the tumor region, or any area of your interest. So you can actually see the spatial data where your immune cells are present in situ in the tissue section. And you can locate where these cells are present in different distance zones, like created in here. And then taking it one step further, what we call it as phenotypic interaction is where you can actually define, after performing the phenotyping, you can actually define one of the phenotype as your source phenotype and the other phenotype as a target phenotype, like a primary phenotype and a secondary phenotype. And you can actually give it a distance, like look for the maybe CD4 PD1 double positive cells next to the epithelial cells. So you can ask such questions, and then what you get as an output are these kind of cell-to-cell interactions where you can define one phenotype of your interest and as a target and the other phenotype as a source, and then you look for the probable cell-to-cell interactions within those zones that you have created. And again, as a similar to the previous sample, coming down to the scatter plots, this is one of the really updated feature in StrataQuest 8 that you can create scatter plots in 3D. So you have the intensity of CD4, intensity of CD8, intensity of CK, all on in the 3D diagram. You can create these scatter plots, you know, you can do the cutoffs of these scatter plots in 3D as well. And similarly, if I just open these diagrams in here, you can look into creating the violent plots. You can look into creating the song, that is a dimension reduction analysis. You can have a CD4 PD1 double positive phenotype, CK or CD8 PD1 double positive phenotype. Phenotypes for T helper cells, you can do phenotyping. You can create UMAP. You can create Disney plots by defining what parameters like different intensities or different, different other measurements that you have done to club different kind of cells or phenotypes together. And all these data can be exported by one click of a button, and then it is ready to be used in your publication. Taking it one step further before I close my presentation, as I mentioned, in this case, it was just 5 markers, but there is no channel limits with StrataQuest. We support analysis for imaging mass cytometry, for RNA scope, and for multiplexing, for multispectral imaging. So one of the sample that I have here is a sample that was stained using the, I think it was Akoya staining. It was, yeah, multiplex phenocycler staining, and it was 37 different markers. And then if I click on the dimensions, just give me a second. You can see there are 37 different markers or different channels present in this particular sample. So when you have a multiple staining round and you have 37, 40, 45, 50, 96 channels, the sample becomes really oversaturated. So with StrataQuest, what you can do is, if once you have a research question ready, you can look into, switching on only the channels that are required and switching off all other channels and do the analysis in those, and then switching back on the next ones that you are required in the next set, and then combine the results of both of them together. And what you can get as an output is, if I can show you, a plethora of data which is ready to go in publication. Like in this case, it was I think melanoma sample. So you can identify how the epithelium thickness is changing, along the sample. You can look into different phenotypes of the markers. You can look into 3D diagrams. You can create gates, as I showed in the earlier sample as well. You can create Disney plots, U maps, again, 3D markers, violin plots, distance analysis, where the leukocytes are present from the epithelium, where cytotoxic T lymphocytes are present in relation to the epithelium, and much, much, much more. So StrataQuest provides you with the tools to analyze innovative new research questions, as well as provides you with the flexibility to interpret those results if any of the manual corrections are required, as well as it provides you with the tools on how to summarize your data. Because in the era of multiplexing, everyone wants to do 97 plex, but they do not know how to analyze those results. So they can do the image analysis for those, but what that data is representing in correlation with each other, that is really difficult for a lot of researchers. So with StrataQuest, we provide all those tools. And with this, I would like to close my presentation, and I'll be more than happy to take any questions. Thank you.
Thank you, Sanchit. I think this was really impressive what you can do with StrataQuest. I
Thank you
think
so
we
much.
can now switch to the Q&A session.
Yeah.
So first question would be, which sample types can I analyze in StrataQuest?
That was a very good question, Anastasia. And I think I have mentioned in the presentation as well that in tissue gnostics, we provide a whole solution for the scanning as well as for analysis. But our StrataQuest platform is standalone as well. So we support most of the third-party, scanners, tissue scanners that are available in the market, be it Olympus, be it Zeiss, be it Akoya. If you have just uh, generic file format like TIFF files, multi-TIFF files, or OME TIFF files. So you can import any of these files and do the analysis how I showed you. And there is no limit on that certain file types can do certain kind of analysis and not the other kind of analysis. You can do analysis for all the images.
I see. Thank you, Sanchit. Okay, let's move to the next question. I have custom analysis pipelines. Can I integrate them within StrataQuest?
So in StrataQuest, as again, I have described that we provide tools for the core facility managers, which they do have their own workflows as well as we provide the custom-made analysis solution for labs, which are very specific for one research question. So in StrataQuest, there is an interface where you can actually perform some of the analysis in StrataQuest, and then send it into that interface, and you can run your own pipeline or whatever pipeline you have created, and then feed back the images in StrataQuest for the rest of the analysis as well.
I see. Thank you. Okay, next question. I use Opal Kit from Akoya. Is it compatible with StrataQuest?
Yes, I get this question a lot that if it is compatible with StrataQuest. We do not put any restrictions on what kind of staining to use. We do not put any restrictions on what markers to use. Basically, if you're using StrataQuest, we just say if you can stain it and scan it, we can analyze it. So we do not put any restrictions on which markers, how many channels. So yes, we are compatible with Opal kits.
Mhm. Moving on. How long does it take it to analyze a tissue section?
So that really depends on the size of the tissue. So if you have a size, if you have just a TMA core or just a size of a tissue, what I have showed you today uh, in the colon sample bright field one, that is literally instantaneously you can do the analysis. And the TMA core, what I showed you, one core that can take few minutes. But if you have a say whole mouse brain, the same workflow can take a little bit longer as well. So it depends on how many field of views it has to analyze.
I understand. Okay. Currently, we have a multi-omics project where we receive data from mirrorfish. Is there
Mhm.
any possibility to analyze or integrate this data into your software?
So we are already working on the mudfish data, not with 8.0, but with 8.1, we are creating an interface where you can actually import your mudfish data on StrataQuest, do the analysis what I showed it to you, and in addition to that, you can actually look at your genomic level data for each single cell within StrataQuest. So we are working on that. We do have the solutions for other multi-omics techniques like imaging mass cytometry. If you are running those, if you have images for those, you can do the analysis using StrataQuest. If you're running RNA scope or any RNA interference techniques, so you can do those analysis for now. But Mudfish is in the pipeline.
That's great. Okay, next question. We have established analysis pipelines in the lab. Can I transfer them to StrataQuest, or do I need to redevelop everything from the scratch?
I think this is related to the one question that we had earlier as well. You cannot import directly into StrataQuest those pipelines. So what you can do is we provide an interface which talks between your script and the StrataQuest engine. So if you use some of the tools within StrataQuest, and then you can put it as an export for your images to go and run your pipeline, and then come back in StrataQuest to do the to continue with the analysis workflow. So it is
Okay.
possible. It is not, you cannot import your pipeline into StrataQuest, but we provide an interface where it can be, talking to the StrataQuest results versus the results that you're getting from your pipeline.
Mhm. Okay, I see. Okay, and we have the last question, I think, if there's nothing else from the audience. I understand TG is based in Europe. How do you provide support to your users on other continents?
So although our headquarters it's in Europe, in Vienna, in Austria, but we have a global structure. So we have our Tissue USA on East and West Coast. We have Tissue China. We have Tissue South America. Like I am from Tissue Australia. So we do provide support on all major, like I would say, yeah, on all continents except Antarctica for now. So yeah, but our support team is always available. Obviously, the representatives of Diagnostics in that particular area, but you can reach out to our support team in Vienna anytime. So it's pretty a cohesive environment where you can have your research questions answered.
I see. Thank you, Sanchit. Thank you,
No
everyone
worries.
who joined our webinar today. And if there are no further questions, then we thank everyone for joining us today, and we hope to see you in the next Tissue Diagnostics webinar. Have still a nice day, and goodbye.
Right. Thank you.